Hello, today we will be discussing current nerve transplant research and how advancements of this topic can aid in human medical advances. Neurons carry impulses from their dendrites through the axons and down to the synaptic cleft to be exchanged. In the transmitting neuron, it will intake calcium ions. This then allows for the neuron to release the necessary enzyme to the target cell. When a neuron is damaged, the proper enzyme can either not be released or cannot be carried throughout the axon. There are 4 different types of nerve cells. The first is a bipolar cell. These have one axon and one dendrite both branching off in separate directions. These are found in the sensory pathways and are used to produce smell, sight, taste, hearing, and vestibular functions. These are very rare within our body. Next are unipolar cells. These have one arm coming off of the cell body. They transmit nerve impulses and are found mostly on the spinal cord. Third, there are multipolar cells, which have a single axon and multiple dendrites. These allow for many messages to be received by the neuron. They're most commonly found on the brain and the spinal cord. Lastly, there are pyramidal cells. These are found in the brain in places such as the hippocampus. These perform cognitive thought processes. They have impulses that run along their long axon, and they're shared by many different dendrites that branch off. Once a nerve is damaged, degeneration can begin within 6 months. Therefore, it is super important to get any necessary surgery to try to repair the nerve as fast as possible. We all know and understand the general concept of a nerve transplant and nerve grafting, which is just the process of switching one area of a nerve with a replacement. However, there are 5 different types of nerve grafts. A nerve autograft uses a nerve from another area on the body of the patient to be a replacement for the damaged nerve. For example, if the patient came in and had nerve damage on their long thoracic nerve, it would be removed and replaced with either a medial pectoral, thoracodorsal, or intercostal nerve. Second is direct repair nerve graft, which is almost the exact same, except it's from a donor. In direct transfer nerve grafting, the damaged nerve is removed. Then the proximal end is directly attached to the primary receptor. An end-to-site sensory nerve graft removes the damaged nerves and reattaches the 2 working ends together. This is the top recommended type of nerve graft when dealing with nerve damage. Lastly, doctors are doing nerve conduits. Doctors will remove the damaged nerve and then replace it with a man-made conduit. The nerve conduit is hollow on the inside, so it allows for regeneration of nerves and provides structure for axon growth. It generally takes 4 weeks to reattach, then the rate of regeneration is about 1 millimeter per day. Here are some examples of the different kinds of nerve grafts. First is an example of the direct repair nerve graft. This shows how it works. The donor nerve is removed from the donor, most commonly from the leg. The damaged nerve is then removed. The donor nerve and remainder of the healthy nerve are reattached using sutures. Next, we will look at an example of an autograft nerve graft. We see in the first photo that surgeons have removed what they consider the damaged portion of the nerve. They then replaced it with the medial antebrachial nerve graft. Lastly, we will take a look at an example of a nerve conduit. When looking at this picture, we can tell this is a conduit because it does not match the surrounding nerves. As I have mentioned before, the conduit is a great alternative because it really helps in the regeneration of nerve growth while still allowing for the electrical stimuli to be passed through. With the spinal cord being an abundance of these nerves, it is much harder to perform a surgery on a full spinal cord. Regeneration and axonal growth would need to happen fast and with complete accuracy. If one nerve is connected wrong, the entire surgery would not work. Due to issues like these, doctors have yet to complete a spinal transplant on human patients. With any sort of transplant, the body could experience some sort of rejection. It could be hyperacute, meaning the organ or nerve is rejected within a few minutes to hours after surgery. With acute rejection, rejection occurs days to weeks after surgery is performed. Lastly, chronic rejection happens years after surgery. When a nerve is rejected, it means our body's own immune system recognizes it as foreign and wants to kill it off. Our body will send antibodies straight to the nerve and break it down. If not treated in time, the nerve will fail and will not work anymore. As well as many other surgeries, nerve grafting will come with possible complications such as hemorrhage, blood clots, and so on. However, with nerve graft, there is a much higher chance of experiencing nerve damage after the surgery. New research done with nerves are not performed on humans due to many people not agreeing with the ethics behind it. The trials are too dangerous to perform on humans, so we test them on animals such as rats, canines, or monkeys. Rats have been used to perform the first American successful head transplant. Here is a video to help explain a little more on how this was performed.
Some scientists want to perform head transplants in humans. Considering we are not there yet, some researchers are experimenting with animals. A group of scientists recently engaged in such an endeavor to identify potential blood flow and rejection problems. The procedure used involved starting with 3 rats to end up with a 2-headed one. Of the original trio, one was the donor and another the recipient. The third was there to ensure there was not an interruption in the blood supply to the donor's brain during surgery. To that end, the head and shoulders of rat number 3 were separated from the rest of its body. The upper portion was then sewn onto the donor and the blood flowed through a tube and into the donor rat's brain. Meanwhile, the main supplies of recipient and donor were becoming one, as their major veins had since been connected. This procedure was performed on a number of trios, and according to the scientists, 14 groups survived for roughly 36 hours.
Another test was done on beagles to test the amount of regeneration after a nerve conduit was placed. The conduit was a cervical sympathetic ganglion block, also known as CSGB. This type of block is known to aid in blood flow. With this study, CSGB was placed into the mandible of the canine. It was found to cause regeneration to happen faster when used as a nerve conduit. It was also found that most nerve function was returned, but not all. We can see in this thermal image that the canines ended up having an abnormal increase in blood flow 10 weeks after surgery. So with the advancement of nerve grafting and neuronal research, we can then dive into the idea of a human head transplant. An Italian neurosurgeon, Dr. Sergio Canavero, has been striving for a few years to perform this amazing surgery. He has a willing volunteer who has a neuromuscular degenerative disease who is willing to have his head transplanted onto a cadaver's body. In the mid-1900s, a Russian surgeon completed what is known as Frankenstein's dog. He successfully transplanted a new upper torso onto the neck of another dog. He performed this kind of transplant on multiple occasions with many canines. Most lived for months at a time after surgery. This ended up fueling a race between Americans and Soviets on medical advances. The issue with this surgery is understandably the ethics behind it, but also certain hormone imbalances and also how to reattach the spinal cord and the abundance of nerves. When talking about the hormone imbalances, we need to think about if we were to transplant a head from a woman to a man. The estrogen levels would be off and the patient would have to worry about medications for hormones as well as everything else. Also, growth factors could pose an issue. If someone still produced any growth hormones and their head was transplanted to someone else, they would potentially start growing again without the body being adapted to it. What we are most worried about is fusing the spinal cord back together. As mentioned before, we have not fully figured out how to reconnect a spinal cord after it has been severed. Dr. Canavero is very confident that he can perform the surgery. He plans on following the steps listed above. Freezing the head and the body first. This will prevent brain cells from dying. Then, the neck, blood vessels, nerves, and spinal cords are cut with an extremely fine knife. This will make reattachment much easier. The heads are transferred and the spinal cord is fused with glue. All other things are sutured back together, and as he said, we shall hope for the best. Dr. Canavero believes he can use polyethylene glycol to fuse the spinal cord back together. PEG has been shown to aid in axonal regeneration. Axons have a hard time regenerating by themselves due to the overabundance of hydrogen peroxide and calcium ions that are present when the nerve is damaged. The PEG gives the membrane an environment which reduces the amount of these ions. It has been found to do this by reforming the cellular membrane, which disrupts the damaged cell cycle. We also see that PEG reduces the amount of mitochondria-derived oxidation stress within the neuron. Researchers in Konkuk University did a study in which 90% of a canine spinal cord was severed. Then they fused it back with PEG. They found that within 3 weeks, the canine was able to wag his tail and walk once again. Advancements like this can help with neuronal cosmetic surgeries. Examples include after strokes, people will have half of their face droop due to the nerve damage. With the right knowledge, we can then cosmetically enhance the face to make it look normal again. More importantly, it would allow us to fix paralysis. With Dr. Canavero starting to venture into PEG's spinal connection, we could try to transplant spinal cords so paralysis patients can perform the necessary actions once again. Overall, we have discussed how nerves work and transmit signals. We learned the difficulties of nerve grafting and spinal cord transplants. We also went over how mice and canine research has helped us grow our knowledge. Lastly, we talked about the infamous human head transplant. I believe if we continued to further our experience with spinal cords and nerve regeneration, we could then complete the spinal cord transplant. I would like to thank everyone for taking the time to listen to this presentation.